This video review is to aid you in preparation for your comprehensive final exam. Your exam will consist of 100 multiple choice questions. The questions will cover only those chapters studied this semester. Students will need to bring a Scantron to the exam. We began our semester by examining the scope of microbiology and some of the history of microbiology. Students should be able to define microbiology and recognize what is studied in the field of microbiology. Students need to be able to provide a description of the groups of organisms studied in the field of microbiology, including the bacteria, archaea, fungi, algae, helminths, and viruses. You will find summaries of the characteristics of those groups on pages 3 through 7 of your textbook. We studied a number of individuals who have made contributions to the field of microbiology this semester. I want you to focus on two individuals, Antony van Leeuwenhoek and Louis Pasteur. The last section of Chapter 1 addresses modern microbiology. You will need to be able to describe what is studied within the field of immunology and epidemiology. Table 1.3 displays the fields of microbiology and the scope of the studies. In Chapter 2, we examined the chemistry of microbiology. You will need to begin by making sure you are familiar with the structure of the atom. This includes knowledge of the three subatomic particles, where they are located within the atom, and their charges. You should also be able to describe how atoms interact to form bonds. This requires that you understand valence shells. You will find useful reading material regarding valence shells on pages 27 and 28 of your textbook. Students should be able to differentiate between covalent, ionic, and hydrogen bonds. Be aware of whether electrons are being shared, equally or unequally, if there is a transfer of electrons, or if the bond is simply due to an attraction related to charges between the two atoms. An understanding of organic molecules is important in any biology course. Students should be able to list the 4 classes of organic molecules, as well as be able to give a description of their characteristics. You will need to be able to identify the monomer for those classes that have one and recognize representatives from each class. You also need to recognize the structure and function of adenosine triphosphate, which can be located on page 51. Chapter 3 is an important chapter. In this chapter, we explored the 2 different types of cells and examined the structural features of each type of cell. Everything we learned after this chapter continued to revolve around cellular structures and their functions. Students must recognize the difference between prokaryotic and eukaryotic cells and recognize what groups of organisms are prokaryotic and which groups are eukaryotic. Students are also expected to recognize the structural features of each type of cell and the function of each of those structures. Table 3.3 summarizes some of the structural features of prokaryotes. Students should be able to easily recognize bacterial cell structures and identify the function of those structures. Please be aware that you are expected to do more than regurgitate a simple definition of the structure. For example, the function of the bacterial cell wall provides support and protection for the cell. We learned significantly more about the cell wall, including its chemical composition, and we differentiated between gram-positive and gram-negative cell walls and discussed important information such as the endotoxin lipid A. So it will be important that you still recognize that type of information. Eukaryotic cells are compartmentalized structures with many organelles. You will be responsible for knowing the organelles and their functions. In addition, recognize that plants, animals, protozoans, algae, and fungi are members of this group, and that there are some differences that exist within the group. For example, only plants and algae have chloroplast. Another example of an important difference is the absence of the cell wall in animals and protozoans. Table 3.5 gives a very summarized view of the non-membranous organelles and membranous organelles of prokaryotes and eukaryotes. And Table 3.6 provides a comparison of bacterial and eukaryotic cells. In addition to learning the structural makeup of the 2 types of cells, we also learned in Chapter 3 how to move materials into and out of those cells. You will be responsible for recognizing what is meant by diffusion, facilitated diffusion, osmosis, group translocation, active transport, endocytosis, and exocytosis. Tables 3.2 and 3.4 in your textbook summarizes these processes. Many of the concepts studied in Chapter 4 were employed in the laboratory component of this course. Students are expected to have an understanding of the purpose of a smear, heat fixation, and staining, as well as explain how they are performed. Students perform two types of staining techniques this semester: simple stains and differential stains. Students should be able to differentiate between the two and explain why each is performed. Students were required to perform 4 different types of differential stains in lab this semester: the gram stain, acid-fast stain, endospore stain, and negative stain. For each of these staining techniques, students should explain why the particular stain is performed. In other words, what information do we gain in performing the stain, and recognize the results of the stain. For example, following the gram stain, a cell observed to be pink in color would be referred to as gram-negative. Pages 105 through 110 addresses the staining techniques, and Table 4.3 provides a nice summary of the staining techniques. Chapter 4 also addressed the classification and identification techniques for microbes. You will need to recognize the 3 domains of life and have a general understanding of how microbes can be identified. In Chapter 5, we explored microbial metabolism. Students will need to be able to define metabolism, catabolism, and anabolism. They will also need to define enzyme and understand the importance of enzymes in regards to metabolism. Students will need to differentiate between the competitive inhibition of an enzyme and non-competitive inhibition of an enzyme. Information regarding the 2 types of inhibition can be found on page 132 of your textbook. A large portion of our time in this chapter was dedicated to carbohydrate catabolism. Students need to differentiate between aerobic cellular respiration, anaerobic cellular respiration, and fermentation. Figure 5.12 provides a visual summary of the differences between respiration and fermentation. Table 5.4 provides a comparison of some of the characteristics of the 3 pathways, including differences in the final electron acceptors and energy yield. In Chapter 6, we examined concepts related to microbial nutrition and growth. We encountered a number of new vocabulary words that are important in the field of microbiology in terms of describing the needs of the organism. The student is expected to know what these terms mean when seen. The terms related to oxygen tolerance include aerobe, anaerobe, facultative anaerobe, microaerophile, and aerotolerant. In addition to recognizing what each of these terms means, you will also need to be able to explain what permits organisms to grow in the presence of oxygen. This referring specifically to the enzymes superoxide dismutase, catalase, and peroxidase. Figure 6.3 illustrates the different oxygen requirements of bacteria and provides a brief description of each. We learned several terms that permit us to recognize the temperature preferences of microorganisms, including psychrophile, mesophile, and thermophile. Other needs we considered included salt tolerance, in which we discussed facultative halophiles, and we examined the term fastidious, referring to microorganisms that required access to organic growth factors. Students should be able to explain what occurs to a cell when it is placed in either a hypotonic or hypertonic environment, and what adaptations or structures permit organisms to survive in such environments. We spent some time discussing how bacteria are grown and isolated in laboratory settings. Students are expected to know the importance of the streak plate technique and describe the different types of media and their functions that were discussed in class, including enriched, selective, differential, reducing, and transport media. Finally, in this chapter, we explored the process by which bacterial cells divide and their growth curve. Students should know what binary fission is, be able to define generation time, and describe the phases of the bacterial growth curve. In Chapter 7, we examined microbial genetics. Students should describe the structure of DNA, including concepts such as components of the nucleotide, the anti-parallel nature of DNA, complementary base pairing, and its helical structure. Students should be able to identify what a plasmid is and list and describe 4 different types of plasmids. You will find a description of each on page 196 of your textbook. We examine 3 different genetic processes in this chapter: replication, transcription, and translation. Students should be able to define replication and have an understanding of the purpose of the process. Students should recognize that transcription and translation are 2 required steps for gene expression. Students should define each process and know what is produced as a result of each. Table 7.3 provides a summary. An important concept that you may have encountered for the first time in this course is that of horizontal gene transfer. It is important for students to recognize that horizontal gene transfer occurs between bacteria and that it contributes to the development of antibacterial resistance. Students should be able to list, define, and describe 3 types of horizontal gene transfer: transformation, transduction, and conjugation. Table 7.6 summarizes the 3 mechanisms of horizontal gene transfer and their requirements.